So, hello and welcome to the Cheeky Science Show, where today I'm joined by Cathy O'Hare, Head of Operations at NewLimit, the pioneering biotech company focused on extending human healthspan through epigenetic reprogramming. In our conversation, we discuss how NewLimit is trying to identify ways to rejuvenate cells and the current progress the company has already made. I hope you enjoy our conversation. So, you're head of operations at NewLimit. So, before we sort of dig into a bit more details about what exactly NewLimit is doing at the sort of operational level, could you give a sort of big overview to NewLimit as a company?
Yeah, so NewLimit is a longevity-focused company. We have decided to focus on partial reprogramming as the technology that we use for drug development, and we can go into the details of that subsequently. We're about 33 people to date based in South San Francisco. And it really came out of the founders, Brian Armstrong, Blake Byers, and Jacob Gimmel, just like couldn't stop thinking about epigenetic reprogramming and how this could be applied to longevity. And so, as you do in the San Francisco Bay Area, if you can't stop thinking about something, you start a company. So
I see. And so, what was your intro to NewLimit, and could you give a sort of overview to your role as head of operations?
Yeah, so my background is as a scientist, a PhD trained as a neuroscientist, made a transition from academia into industry about 10 years ago, started my career as a biomarker scientist in translational medicine, worked my way into the clinic, and ended up running US operations for a diagnostics company. I like to say that I am a scientist by training, but generalist by nature. So I have, over the course of my career, just sort of gravitated towards more and more generalist roles. But my background is really in drug development and was most recently at GSK running a clinical stage liver disease program. And I got introduced to NewLimit about a year ago and was intrigued. I had never really thought that much about longevity. I liked the approach. I think it's unique to find a company that's this ambitious, but also this sort of grounded in like near-term value generation and near-term scientific development. And that's part of what piqued my interest. And I sort of had a light bulb moment in talking with the founders and the rest of the team here. In drug development, we've spent years looking for genetic targets for disease and environmental targets for disease. And we've largely neglected age as a driver of disease, even though it's kind of the only thing that underlies basically every major disease. And we just never really thought about targeting it as a driver of disease. And that's basically what NewLimit is doing, is targeting cellular aging as potentially the root cause of a number of diseases. And if you add up all of those various diseases, if you can treat them, you end up adding years to the human health span.
Exactly. And so you mentioned earlier this term partial reprogramming. So could you explain a bit more? What exactly does that phrase mean to you or to the company?
Yeah. So partial reprogramming at its heart is trying to reprogram the age of cells without changing their cell type. So we know through work by Dr. Yamanaka that you can take old cells and turn them into stem cells. iPSCs, or induced pluripotent stem cells, are taking an old cell and inducing them back into a pluripotent stem cell state where they could turn into any type of cell, right? We all started as one cell, and then that one cell divided, and then different cells became different cell types, and that's how we ended up as a fully formed human. So we know from that work that with just 4 transcription factors, you can change the age and the type of a cell. There were subsequent experiments that were done to show that you can change the type of a cell without changing the age. So you can take an old skin cell and you can turn it into an old neuron, which is pretty incredible because actually to go from being a skin cell to a neuron that's firing action potential seems huge, but you can do with only sort of 6 transcription factors. So we know that cell type and cell age can be dissociated. What hadn't been tried yet is, okay, can we keep the cell type the same, but just change the age? So take an old hepatocyte and turn it into a young hepatocyte. So when we talk about partial reprogramming, we're not reprogramming the cell all the way back to its stem cell state. We're just partially reprogramming it back to a younger version of itself while maintaining the cell type and the functions that are characteristic of that cell type.
I see. I understand. And so you mentioned Yamanaka's sort of landmark paper, and that, of course, had 4 different transcription factors that was expressed to convert the cells back. And so I believe, or at least my understanding of NewLimit is you're also trying to identify transcription factors that can do this partial reprogramming. So could you tell us about the sort of search process or how you go about doing this?
Yeah, so we've built everything from the ground up. We have an in-house lab and we've built a discovery funnel and a screening engine. And what we do is we screen for transcription factors that rejuvenate or sort of change the cellular age of cells. The first thing that we did was to bring in samples from old and young donors for hepatocytes and for T cells and try to figure out what is the difference between an old cell and a young cell. So what does it mean to be an old cell, and is that something that we can characterize? And so we built an age inference score based on gene expression to look at basically what is the gene expression profile of a young cell and the gene expression profile of an old cell, and that's how we define old versus young. The next step then is to find transcription factors that will change that score, right? So can we get a cell from an old donor and express transcription factors that make it look like a young cell? And when we say look like a young cell, we mean give it the gene expression profile of a young cell. So that's the first step in the sort of wet lab screening funnel. Once we get hits from that screen, we then move on to functional assays, because it's great to make an old hepatocyte look like a young hepatocyte, but if it doesn't act like a young hepatocyte, it's not actually helping anyone or any disease state. So we then take those hits into functional assays. Based on the cell type, they're different. So hepatocytes have different functions from T cells, but we've identified a number of functional assays where young cells are much better at that function than old cells. And then we take our hits out of the initial screening funnel, we express those transcription factors in the old cells and see if we can get them to act like young cells. So for hepatocytes, it might be proliferation or regeneration after a liver resection. It might be metabolism of certain different substances, metabolism of alcohol. I mean, we've all sort of experienced the fact that as you get older, your liver's not as good at breaking down caffeine and alcohol as it might have been when you were younger. And so we see the same thing in these assays that we run in mice.
I see. And so actually you've raised several questions at the moment for myself. And so to go back to this sort of search identification for transcription factors, I should probably know better because I'm a biochemist how many transcription factors we have in our genome. But of course, it's not just about putting one into the cell. It could be the different combinations. So, and that's when you think about if you put A with B and C, and then you have all these like huge numbers of possibilities. And so how is NewLimit overcoming this actual like massive search that it has to do?
Yeah, so you raise a great point, which is the search space for this is massive. So even if we aren't looking at every transcription factor that exists, even if we're just sort of making our best guess at what are the transcription factors that are likely to be involved in cellular aging, and say we were to pick 100 of them, once you start doing combinations, you end up with an enormous list of all the different combinations of all the different transcription factors, and it would be impossible for us to sort of brute force our way through that. So actually, what I didn't talk about before we get into the wet lab is the in silico screening that we do beforehand. So we have built a machine learning based model that ingests all of the data. We started with publicly available data. We then we've screened millions of cells at this point. And all of that data goes into our model. And this in silico screening basically predicts what transcription factors we should test in the wet lab. And it gets better the more data that we ingest. So we've screened 14,000 transcription factors at this point, which is orders of magnitude more than the entire field had screened before NewLimit became a thing. And because we have such a large data corpus, we can keep feeding that data in and our model just gets better and better. And the nice thing about the model is it doesn't have to be perfect, right? We're not trying to build a model that can predict what transcription factors will work in the clinic and then take them into the clinic. It just has to be directionally correct. It just has to give us a sense of, cutting it down from 100 to a smaller number that we can actually work with, and we can run those combinations so we can take them into the clinic and then take them into a wet lab screen. And then as we test the hits that come out of the in silico model, we then put that data back into the model to say whether it was right or wrong, and it gets better with every iteration.
Okay, I see. And so in terms of when you add these genes to the cells, are you doing sort of temporal exposure to these different genes, or are they actually integrating, or how is your system?
It's a transient expression. So we express mRNA. Ultimately, the drugs that we develop will be mRNAs encapsulated lipid nanoparticles. And so this is not gene therapy. There's no change to the DNA. We're not integrating to the DNA. We are just expressing mRNA transiently. One of the nice things about epigenetic reprogramming, if you think about the way it works though, epigenetic programming is what made different cell types be a certain cell type, right? And that cell type has persisted for the duration of your living as a human. And so we know that epigenetic changes can yield durable effects. And so we're hopeful that with this transient expression of mRNA, if we find the right transcription factors, that effect could be durable. That's not to say you'd only have to dose one time, but we're hopeful that it would be relatively infrequent dosing and you would get these lasting effects because you're not just turning on a signaling pathway, you're actually trying to reprogram the cell back to a younger state.
Exactly. So that kind of makes sense to me. So I guess it's even more impressive as well because you say you only sort of have this short time window in which you get the expression of these transcription factors. And so I think one question that I've always had, and I'm curious to know how long they do sort of persist in the cell before they get degraded. How long do you think it takes to rejuvenate a cell?
So I'm trying to think if I know the right answer to that. So we've done experiments where you turn them on and then a few days later we test and we see the effect. So the sort of onset of the potential therapeutic effect is relatively quick. We've also done experiments where we turn it on, we then chase it, turn it off, and a week later we test, and these effects are still durable. So I think we're hopeful that we could get relatively quick onset and a durable effect with some of these.
I think, and also from a safety perspective, I think there's still concerns about the more traditional virus-based approaches that if they have chance of integrating into the genome, that could have safety consequences. And the fact that the mRNA can get degraded but still have this really immediate effect is very impressive and, of course, important.
Yeah, there's no risk of integration. The mRNA can't integrate. And, one of the reasons we've chosen LNPs or lipid nanoparticles as our delivery method is because these are already commercially available, right? So the safety of delivering a drug via lipid nanoparticle has already been established, at least for the lipid nanoparticle itself. There are drugs on the market today that use LNPs for delivery. That makes hepatocytes a very obvious choice for one of our programs because most LNPs go to the liver. And so that's where the commercial applications have already been established. There is a lot of work in the field to develop LNPs that will target to other organs, so to T cells, to endothelial cells. I think we're hopeful that there are other groups working specifically on LNP targeting. And so as we onboard more and more cell programs, the technology will come along such that we can target specifically to those cell types.
I see. Yeah, that makes sense. And so you've said at the moment that you're targeting hepatocytes, and I know you've also been looking at T cells and these factors that you've identified that have already had success. Compared to the original sort of Yamanaka where there was 4 factors, how many factors are you sort of aiming for in terms of the number of transcription factors?
So it's a mix. So some of the hits we have are a single transcription factor, some of them are 2 or 3. You could probably package 5 or 6 within a lipid nanoparticle. It's just a matter of whether you would need to. We don't have a strong preference on like, oh, it has to be a single TF, it has to be 3 or more. I think we're looking for the right approach. So yeah, at this stage, it's a mix. Some are single, some are a combination of transcription factors. And I expect we'll see the same thing across cell types.
Okay. And I guess, do you expect that these transcription factors are going to be different depending on the original cell type? Or do you think there will be some sort of like universal, the best transcription factors?
I think we would love it if there's something universal. We're not at this stage yet where we've tested across our programs, right? So we haven't really taken hits from our metabolism program in hepatocytes and tested it in T cells. We're still in the stages of sort of identifying hits on each of those individual programs. I think it would be great if there was something that was universal. The LNP targeting will be different though, right? So even if you found one TF that like this was the TF for every cell type, whether there'll be an LNP that you could use that could get it to every cell type, whether you would want that sort of non-specificity, I'm not sure. So at this point, we don't have an answer to that. And yeah, I think that the verdict is still out on whether that's where this will end up.
Yeah, and I understand that it's even impressive how much progress NewLimit has made in such a short space of time. But I understand that you cannot like reveal exactly the transcription factors that you've identified at the moment. Yeah. But has there been any sort of biological surprises or things that have surprised you about your discoveries?
There definitely are hits that have come out of our screening funnel that you wouldn't have otherwise suspected just from reading the literature, right? So some things have come out of our screening funnel that might have been anticipated based on the biology, which is really nice when you're first building a platform because it gives you some validation that what's coming out of your funnel actually makes sense from a biological standpoint. The hits that we're progressing now are completely novel and haven't been linked to epigenetic reprogramming, haven't been linked to longevity, haven't been linked to any disease. And I think that's really where we want to play. That's the strength of the platform, is it gives you something that you couldn't otherwise have just found by reading the literature. So there definitely have been surprises on that front.
Yeah, exactly. No, that completely makes sense. And it justifies, your approach of doing this really broad search.
Yeah. And you like, ultimately you want to mix, right? You want some things to come out that you're like, oh, that makes sense, because then it suggests that your platform's right. And then you want a bunch of stuff that you would never have found if you hadn't built that platform. And I think we're very fortunate to be in a position where we've sort of ticked both those boxes.
Exactly. And so my understanding is now that you've found some potential transcription factors, you're starting to begin or start to plan preclinical studies. But I also wanted to quickly mention, because I did read and understand that you're doing in vivo screens as well as sort of like the traditional in culture screens. And could you elaborate a bit about that?
Yeah, so particularly on our hepatocyte program, our metabolism program, we do our screening in a humanized mouse. And we believe this is the first time this humanized mouse model has been used for pooled screening. And effectively, there are transgenic mice that exist that they have to be supplemented with a substance in the water supply or in their food to keep the hepatocytes alive. And if you take that away, the hepatocytes die. And what you can do is you can inject human hepatocytes into those mice while their hepatocytes are dying. And the human hepatocytes will engraft into the mouse liver, and you will end up with a mouse that has a human liver inside of it, which is pretty incredible. It's a very complex model. Marcus Grompe, who's on our scientific advisory board, really pioneered this model. And I guess because NewLimit doesn't like to do anything simple or straightforward, we decided to use this humanized model to do our screening to get sort of maximum relevance for our hits. So the hits that are coming out of even just our initial pooled screening are in primary human cells that are in a mouse liver, so in vivo. And as you can imagine, young cells are better at engrafting into the liver than old cells.
Oh, I see.
And so if you take a mouse, let its liver die, engraft a mix of old and young hepatocytes, the young hepatocytes will mostly take over the liver. So then what you can do is you can express transcription factors in old cells, engraft those into the liver, and see which transcription factors make the old cells better at engrafting into the liver than the other old cells, and compare it to the level of engraftment. And you can even sort of mix multiple TFs together. Each hepatocyte takes up a different mix of them. And then you can demultiplex them, and you can really understand which cells ended up being better at engrafting, what transcription factors or combinations were expressed in them. And those are the initial hits that come out of our screening funnel for hepatocytes. So it's a long experiment. It's complicated, but it means the things that come out of it are so much more relevant than we would get from an immortalized cell line or HepG2 cells or something like that.
No, exactly. No, it sounds really cool, actually. And no, I think, having that in vivo model then places that selection pressure for exactly what you're looking for.
Yeah.
So it completely makes sense.
Totally. Yeah. And we're sort of fortunate that we're looking at overall function and resilience and regenerative capacity of the hepatocytes. And so the fact that the old cells are bad at engrafting is like exactly what we need, because then we can try to make it better. And the challenges of engrafting the cells into the liver serve as our screening funnel.
Exactly. And so I understand that hepatocytes, and that, of course, you have the combination of this great sort of humanized mouse model to look at the liver, as well as the LNPs being sort of the liver's the easiest, I mean, currently, to my understanding, the easiest way to target LNPs is to the liver.
Definitely.
And so therefore, in terms of translating to trying to find a sort of longevity therapeutic, what are the sort of stepping stones that need to be taken next?
Yeah, so we still have a long ways to go, right, in terms of getting to a medicine. As you pointed out, hepatocytes are a great choice for a couple of reasons. One, we have this model we can use. Two is targeting. The other really nice thing about hepatocytes is they have already given us a genetic proof point that perturbations only in hepatocytes are sufficient to impact disease. Because that's a question we get all the time, right? Is, okay, you're going to change one cell type in this organ. That's not going to be enough. Organs are complex. They have multiple cell types, and diseases involve multiple cell types, which is entirely true. But if you look at liver disease, there are risk variants like PNPLA3 or protective variants, one in a gene called HSD17B13. And those genes are only expressed in hepatocytes. And so there are patients that have mutations in those genes, and that is a hepatocyte-specific perturbation, and they drastically shift the risk profile and the course of liver disease. And so there are multiple reasons that hepatocytes as a cell are a good approach and make us feel confident that if we find something that impacts hepatocytes, we can progress into the clinic. So with all of that, I will answer the question you actually asked, which is, yes, we're at the stage of going to preclinical models. So what's next, right? We have hits that have come out of our looks like screen. They've passed the acts like screen, functional assays, and now they're in preclinical models. And so a lot of preclinical models for liver disease are challenging, but there are some that we're using. So we'll test these transcription factors as LNP medicines. So they are actually formulated as LNP medicines in these preclinical models for things like steatotic liver disease, or what we used to call fatty liver disease, and alcohol-related liver disease. And if we see that these potential medicines impact the phenotype in these disease models, we'll then declare one as a lead, and we'll move into lead optimization. What goes into lead optimization is saying, okay, we know what we're targeting, we know what the transcription factors are. We now have to make the best possible version of that medicine, right? We have to find the right LNPs, the right mix, the right size. You know, there are multiple LNPs that target to hepatocytes, so we need to pick the one that works best for us. We need to pick a dose. We need to understand, you know, how the questions you asked earlier, how quickly will the effect take place and how long will it last? We need to understand the PK of this in animals. And basically it's all focused around like, okay, this is what we want to target. This is how we want to do it. We have to make the best possible version of this drug, because at the end of lead optimization, that's the specific drug that we're going to test in large animal models and IND enabling studies and hopefully take into the clinic. Once we have that drug candidate that we feel like is the best version of LNP encapsulating the mRNA for transcription factors X, Y, and Z, we will trigger IND enabling studies. And that's really, you know, there's a very clear checklist from the regulators of this is what you have to prove before you can put this experimental drug into humans. So you have to show that it's safe in a rodent model and a non-rodent model. You have to do very specific tox studies for the duration that you want to dose it in humans. And so we'll move into all of those, and we'll start clinical scale manufacturing, which is understandably regulated in a very different way than the LNP mRNAs that we can inject into mice. And then hopefully we will make it through all of those IND enabling studies with no safety or tox flags, and we'll start a first in human study.
Oh, awesome. Yes, I mean, of course, the LNP, with the fact that it's non-integrative, it's a very small dose, that I think it's already sort of minimizing for the potential for there to be sort of off-target or sort of unfortunate consequences. But I think just more generally, and I think this is like a shared feeling across the whole longevity space regarding these sort of current frameworks that we have in place to sort of test it against certain diseases. If you think that you're then using your therapeutic to try and fix a certain type of liver disease, are you worried that perhaps you'll end up selecting for the best target that works for this disease, but not for aging?
Yeah, it's a great point. And one of the way that we've protected against that is that we've built everything from the beginning of NewLimit to focus on aging, right? So our approach is not, let's focus on disease and let's expand it to aging. Our focus has been, let's figure out what cellular aging is. What is the difference between a young cell and an old cell? Let's treat cellular aging, and then let's find an indication that we can use as an initial proof point. So even if you think about how we set up our screen, right, the first thing that we did was not to say, okay, we're going to get healthy liver cells and diseased liver cells and figure out the difference between them and try to make the disease cells like the healthy cells. No, we took healthy donors, no diagnosed liver disease, young versus old, and we tried to figure out what is the difference between young and old. So from the beginning, our perturbation has been turn the old cell into a young cell. Longevity is not an established indication, right? We can't just launch a trial in longevity right now. We have to have some indication that serves as an initial proof of concept, initial clinical benefit as a stepping stone, but it is start with aging, use disease as an initial proof point that then launches something that's expanded across a larger patient population. So I think that's what we've done to try to protect against exactly the scenario that you've outlined.
I see. Yeah. No, I mean, because my background is purely, academic. And so for me, it's always fascinating to sort of learn about the biotech applications. But of course, I always I'm not scared, but I think it must be very overwhelming to hear all of the different hurdles that you must face to actually go from the initial idea through to therapeutic. But of course, for obvious reasons, to make sure it's safe.
Make sure it's safe. Make sure that you're treating an unmet need, right? Because that's how you get traction. I said this at the beginning, right? One of the things that attracted me to NewLimit was this broad ambition. You can work on a moonshot, but you can also make incremental scientific progress every day. Even if we never make it to a longevity drug, which I hope that we do, that's the goal. Each individual therapeutic area that we pursue is in and of itself a global public health concern and a huge unmet need, right? And that's part of what we think about when we pick a cell type. It's not just, we didn't just pick hepatocytes because you can target to them. We picked hepatocytes because you can target to them. And we think, from a feasibility standpoint, we can identify transcription factors because they're well-established functions in hepatocytes, and because there are huge unmet needs in liver disease. Liver disease accounts for 4% of global deaths, right? There is a huge need for drugs for chronic liver disease. It's one of the major killers that's continuing to increase while we're finding therapeutics for cardiovascular disease and oncology. And so even if what comes out of this is a drug to treat alcohol-related liver disease, that is the stepping stone to what we want to do, but in and of itself is an enormous public health benefit and a huge unmet need.
Oh, no, absolutely. And so, I think no matter what happens, as you say, it would be even incredible to have a sort of very novel, very exciting therapeutic that works for liver disease.
Yeah. And honestly, if you took a transformative treatment for liver disease, a transformative treatment for infectious disease, a transformative treatment for kidney disease, and mix them all together, you would add years to the human health span.
Exactly. Yes.
Yeah.
But I guess thinking more super long term, and if you get this therapeutic through successfully and it works for liver disease, and let's say that you can also now target aging as a disease that's been accepted and that the framework's changed. How do you foresee having this therapy as a sort of longevity therapeutic? And by just rejuvenating the liver, is that going to be sufficient to rejuvenate the whole body? Or how do you consider targeting different organs in the longevity space?
So, I think rejuvenating the liver in and of itself would be huge. The liver performs so many functions and overall metabolic health, right? Your liver is so critical for overall metabolic health. And there are so many people that suffer negative cardiovascular outcomes because of metabolic syndrome. And if you could rejuvenate the liver, I think you could treat something as broad as metabolic syndrome. And so right there, you're adding years to the human health span. Infectious disease, right? As you get older, you are more likely to catch an infectious disease, and you are more likely to suffer negative consequences. So you could envision a drug that is effectively an agent agnostic vaccine, right? So imagine a world where instead of having to get vaccinations for whatever the current strain of the flu is, whatever the current strain of COVID is, whatever, is it RSV this year? What's the infectious agent? If there were a treatment that generally boosted immune resilience, that would be incredibly impactful. Now, to your point, how do you get from treating a specific disease to something that broad? We look a little bit to the GLP-1s as inspiration. So the GLP-1s were initially developed for diabetes, then obesity, now liver disease. Now they're being investigated across a number of therapeutic indications. And we are to the point now where people who do not have an established disease take Ozempic. And that's one way to start to expand. I think our hope is that this becomes widely accessible. What I don't want to do is just make, drugs that no one can get access to. But it's also one of the reasons we've chosen LNP mRNAs. They're very affordable to manufacture. So I think what we would like to see is the regulatory landscape shift. If we can show benefit across a number of therapeutic indications, the hope is we start to move the needle on how you run a trial, how you expand a patient population, what your claims are, right? If you think about getting a drug approved, what you're asking the FDA to give you marketing authorization. You're asking them for permission to make claims about your drug. And so how do you engage with regulators to understand what claims you can make for a drug that has shown benefit across a number of organ systems and that has, helped people live more years and better years?
Exactly. I think it's always a long process, but there's so much that can be found out along the way that's informative for then changing the approach and informing using the information you learn to then adjust the plan. Exactly.
You need data, right?
Yeah.
Like you have to generate data. No one's gonna shift their thinking until we start generating data. And so the best way to start generating data is in disease indications. And then once you have that data, you're in a much better position to discuss, how you can progress this drug, what patient populations you can take it into, and where actually the benefit will be for society.
Exactly. And so I'm going to sort of shift slightly in terms of, at the moment we spent a lot of time talking about the liver. I know your background's more neuroscience. So I think I just wanted to first have a interesting question just to see what are the current ways in which we could potentially target the brain as a sort of rejuvenation approaches, just in terms of delivery modalities, etc.
Yeah. I am sadly a neuroscientist who has not worked on neuroscience for over a decade. That's sometimes what happens when you leave academia and you transition into industry. You go where the opportunities are. And for me, the opportunities have been in liver disease. I would love to see us target something in the brain. It's just such a risky area.
True.
And for us to take that on this early in NewLimit's life, before we've really been able to validate our platform and show that if you identify transcription factors, you can move them into the clinic, it's just hard for us, right? It's really just that technical risk, right? The animal models are challenging. The translation from animals to humans is so unclear. The endpoints for a lot of these diseases in neuroscience are difficult, low resolution, subject to placebo response. And so it's not in any way a lack of desire to target neurodegenerative diseases, neuropsychiatric diseases, whatever we think is most appropriate. It's really just a technical risk that doesn't make sense for us to take on as our, second, third, fourth cell type program. But I promise that I am internally advocating every day for either a neuronal cell type or a glial cell type to be one of the programs that we launch. I just, it's just not yet.
Okay, yeah, understandable. But speaking of the sort of differences between academia and industry, I mean, for myself, I've only really had experience in academia. So would you be able to discuss a bit about the differences that you found?
Yeah, I think at the time that I was in grad school, the mentality really was you can only do real science if you stay in academia. Like that's what I was taught, that's what I was told. That's how we all felt. Having spent 10 plus years in industry now, I can emphatically say that that is incorrect and untrue. I've done more science in my industry career than I ever did in academia, because in academia, I was a one-woman show, right? So as a postdoc, I could do as many experiments as I could physically do at the bench. Whereas in industry, I led a team. I worked with CROs. I had a budget. I ran multiple experiments in parallel. The challenge in industry is the value drivers are different and the focus is different, right? If I'd stayed in academia, I would still be working on neuroscience. I moved to industry and I work on liver disease now. You end up having to go where there's a commercial opportunity or where there's some value driver or some company that's doing it. The big difference, I would say, and this is the advice that I give everyone that's considering leaving academia, is there's a tendency when you're in academia to think of it as comparing two jobs, an academic job or an industry job. It's not comparing one job to another job. It's comparing one job to 1,000 different jobs. The academic track is, I'm not saying everyone's job in academia is the same, but if you're in, get a PhD, do a postdoc, the track that you're aiming for is to be a professor, right? That is one job. And it has probably, it probably feels very different on a day-to-day basis, but it is one job. In industry, there are 1,000 different jobs that have all different types of, day-to-day work, types of companies you can work in. I know people with PhDs that went into project management, that went into business development, that went into operations, that do digital health therapeutics, that do that work in more on sort of, I don't want to say the payer side, but like infrastructure, healthcare infrastructure side. People that work as scientists still at the bench, people that lead a team, people that run clinical trials. Like I'm not a physician, but I ended up in clinical research. So you have to not think of it as one job versus one job. It's one job versus 1,000 jobs. And whatever it is that you want to do with your scientific background, that job probably exists somewhere in industry.
No, you make a good case for it.
That's my pitch.
Thank you.
It's not the dark side. You can still, I promise you can still do real science. Yeah, I remember so clear. I think things have gotten better now. There's much more exposure to people in academia, of people in academia to industry. That wasn't really the case when I was in grad school, but I'm glad to see that's changing to some extent.
No, I agree. But you mentioned this word real science, and for me, I guess real science is beyond just doing the lab work, but also the aspects of publishing and talking about your research. And I think for me, I mean, sort of naively with companies, they often, because they're so protecting of their information for obvious reasons, that it feels like I would feel more trapped, so I can't speak about my research. But I know NewLimit has their blog, and I was just curious to know about the communication side of talking about your research. And are you considering writing papers as NewLimit to sort of discuss your approach and findings?
NewLimit's very transparent. So we share a lot more details about the work that we do than most companies. We have a publication strategy. Yes, there are companies that do not publish, right? There are also labs that do not publish, right? So every lab is different. Every company is different. I think you will find, you just have to seek out the right lab, right? If that's what's important to you is really the science and being able to give lecture or give talks at conferences and publish, then find a company that's one of their core values. If that's not important to you, then don't worry about it. There are labs that are very transparent with the work that they do, and there are labs that pit individual postdocs against each other and they don't even tell each other what programs they're working on. So I think the spectrum of labs is vast, the spectrum of companies is vast. You just have to find one that like fits with your values. NewLimit's made a decision to be very transparent. I think one of the things that we're seeking to do is to be a legitimacy signal in the longevity space. it is a space that at times has, I think it gets criticized of not being a legitimate science. I don't think that's the case anymore, but there's a little bit of, when you tell people longevity, they're like, oh, what are you doing? Like, what supplements are you taking? And you're like, no, this is a legitimate scientific endeavor to understand cellular aging. And we can't be a legitimacy signal in that space unless we share what we're doing. And that's, I think, a big reason why we're so transparent.
Exactly. I completely share the same values. I think that the more that the public can understand what you're doing, the more they're likely to trust you and therefore understand that longevity is actually doing actual research or real science, as you call it.
Yeah. Like, do you want the last 10 years of your life to be better quality? Yes. Okay. Well, that's what we're trying to do.
Exactly. And so to sort of end on a optimistic and note, I want to just ask, how do you envision NewLimit changing over the next 5 to 10 years in terms of scaling the company, the projects, etc.?
Yeah, I think we have no desire to be a single asset company, right? The goal is to be an integrated therapeutics company. So one thing that we're not going to do is shut down our discovery engine. A lot of biotechs, once they declare a lead, they will put all of their energy into developing that lead and sort of shut down the discovery engine. That's not what we want to do, number one, because that's a big risk, right? Because not every drug makes it to the clinic. And two, because if we want to impact overall human health span, we will need multiple assets across multiple organ systems. And so we have to keep discovering more. The biggest change will just be the scope of the work that we do, because transitioning from discovery experiments to preclinical and then into the clinic is a huge leap. It's a huge leap in thinking. It's a slightly different expertise. It's regulated in a very different way for obvious and totally makes sense reasons. We're also not a hyper growth company though, so we will scale, but we're very intentional about being capital efficient, helping our teams be as efficient as possible, and growing at a rate that makes sense for us. So we will, 5 to 10 years, we will have scaled significantly. We will hopefully have multiple drugs in the clinic. How many of those we're developing internally versus ones that we've partnered, I think is still an open question. We will have raised a lot more money, because if you want to run multiple clinical trials, you need a lot more than we raised in our Series B. So I think we'll be wealthier as a company, we'll be larger as a company, and we'll be running multiple clinical trials.
Nice. Yeah, well, I am super excited to follow the journey and just to hear more about it. I think it's already been incredible to watch just how fast the company has grown, and the amount of data you've already collected is incredible to see.
It's quite staggering, honestly. I think when I first came to visit NewLimit, there was a little bit in the back of my mind of like, this is too good to be true. Like, is this real? And then you realize, actually, yes, the funnel that has been set up and the discovery engine that we have is cranking through transcription factors at such a rate that it's just building on itself. And it's actually staggering the amount of progress the company's made in such a short time. So.
Yeah, it's really exciting. And yeah, it's been great to talk to you today, Cathy, and just hear about the insides of NewLimit. And I'm sure my viewers have also found it fascinating. So yeah, just best of luck with everything. And I look forward to finding and hearing more about NewLimit.
Great. Thank you so much.